How Gregor Mendel discovered his theory and benefited scientists. Gregor Mendel, through his work on pre-plant, discovered the fundamental law of inheritance. He deduced that genes come in pair and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant and recessive traits. He recognized the mathematical patterns of inheritance from one generation to the next. Hugo de Vries, Gauss Correns, and Erich Vennesmark each independently duplicated Mendel's experiment and result in 1900, finding out after the fact illegal that both the data and the general theory had been published in 1866, but Mendel questions arose about the validity of the claim that the trio of botanists were not aware of Mendel's probable result, but they soon did credit Mendel with parity. Even then, however, his work was often marginalized by Darwinians who claimed that his findings were irrelevant to the theory of evolution. As genetic theory continued to develop, the relevance of Mendel's work fell in and out of favor, but his research and theory are considered fundamental to any understanding of the field, and he is thus considered the father of modern genetics. How Mendel's theory is applied to meiosis and how the meiosis process takes place. Mendel's law of segregation states that a diploid organism passes a random selected allele from a trait to its offspring, such that the offspring receive one allele from each parent. Meiosis 1. During meiosis, the pair of homologous chromosomes are divided in half to form haploid cells, and this separation or assortment of homologous chromosomes is random. This means that all the maternal chromosome will not separated into one cell, while all the paternal chromosomes are separated into another. Instead, after meiosis occurs, each haploid cell contains a mixture of genes from the organism mothers and fathers. Why chromosomes are important in meiosis? Because first, haploid gamete produced, second, crossing over occur during prophase 1, form, third, homologous pair associate as bivalent and line up at the equator in metaphase 1. Next, homologous pair separate in anaphase 1, sister chromatids separate in anaphase 2, and it is in two divisions. Explain the crossover process during meiosis and its effect on organism. Crossing over is simply the exchange of genetic material between two homologous chromosomes to give rise to recombinant chromosome. In prophase 1, homologous chromosomes align lengthwise or pair with each other. An exchange of genetic material between two chromosomes takes place, which is known as crossing over. The pairing of the homologous chromosome is known as synesis, and the point at which these chromosomes pair with each other is known as chiasma. This genetic variation is required to increase the ability of population to survive. A greater genetic diversity will reduce the chances of inheritance of deleterious traits in the population and therefore help increase the fitness of the individual of a population. An increased genetic variation will also mean a greater variation in susceptibility to disease. So, if there were to be an epidemic of a disease, this variability would prevent the whole population from being wiped out. Another benefit of genetic variation is that some traits that will increase an individual's ability to survive may be introduced in the population.